Good afternoon to all of you. Good afternoon to all of you. नमस्कार. ये कार्यक्रम हिंदी राजभाषा हिंदी एकक की तरफ से आयोजित है. So this program is organized on behalf of the official language unit of IISc. I welcome you all. आने के लिए बहुत बहुत धन्यवाद. आज के कार्यक्रम के लिए हम लोगों ने तय किया कि श्री डॉक्टर अनुराग वार्ष्णेय जी को आमंत्रित किया जाए. इनका यहाँ आने के लिए बहुत बहुत धन्यवाद. आज आप जो है अपने आयुर्वेद के रिसर्च के बारे में हम लोगों को थोड़ा बताएंगे क्या क्या कर रहे हैं. मैं श्री अनुराग वार्ष्णेय जी से रिक्वेस्ट करता हूँ कि यहाँ आएँ, थोड़ा पुष्प गुच्छ और बुके से आपका हम स्वागत करें सबके सामने. धन्यवाद. कार्यक्रम की इस व्याख्यान की अध्यक्षता के लिए मैं प्रोफेसर उपेंद्र नागाथोमा को आमंत्रित करता हूं कि वो इस व्याख्यान की अध्यक्षता करें. आई रिक्वेस्ट प्रोफेसर उपेंद्र नागाथोमा टू चेयर दिस टॉक एंड ब्रीफली इंट्रोड्यूस द स्पीकर.
Good afternoon all. Let me start with English because it's long time since I have spoken in Hindi. आप सबको मेरी ओर से भी नमस्कार. मैं अनुराग को उसकी PhD के समय से जानता हूँ और मुझे बहुत I'm really happy, कि मैं अनुराग को संक्षेप में उसका आप लोगों को पहचान करा देता हूँ, परिचित कराता हूँ. डॉक्टर अनुराग वार्ष्णेय वर्तमान में पतंजलि अनुसंधान संस्थान के उपाध्यक्ष है. वे औषध अनुसंधान एवं विकास कार्य का नेतृत्व कर रहे हैं. डॉक्टर वार्ष्णेय का 22 वर्ष से जीव विज्ञान में अनुसंधान और शोध प्रबंधन में अनुभव है. वे NCBS नेशनल सेंटर फॉर बायोलॉजिकल साइंसेस TIFR बेंगलोर से PhD की. येल यूनिवर्सिटी स्कूल ऑफ मेडिसिन न्यू हार्वेल US में उसने पोस्ट डॉक्टरल की. उसके बाद अपने कार्य के लिए डॉक्टर वार्ष्णेय को रॉयल सोसाइटी ऑफ बायोलॉजी UK का फेलो भी चुना गया है. डॉक्टर वार्ष्णेय जवाहरलाल नेहरू विश्वविद्यालय में एडजंक्ट फैकल्टी हैं. अभी पतंजलि में शामिल होने से पहले डॉक्टर वार्ष्णेय ने समुद्री वैज्ञानिक प्रयोगशालाओं में विभिन्न क्षमताओं में काम किया है. वे रेनबक्सी रिसर्च लैब्स और डाइची सांख्यो फार्मास्यूटिकल्स में काम कर चुके हैं. वे 50 साल से अधिक विविध अनुसंधान विषयों का निर्देशन कर चुके हैं. सो आपका अभिवादन एंड प्लीज अनुराग.
सो प्रणाम, नमस्ते और गुड आफ्टरनून. ये मेरा प्रिविलेज है, ये मेरा एक अधिकार है कि मैं आज उसी संस्थान में वापस खड़ा हूँ जहाँ से 20 साल पहले मैं कभी गया था. तो मेरा शुरुआत NCBS से हुआ और वो इसी कैंपस में यहाँ से ही कुछ दूरी पे है. और जब NCBS यहाँ से शिफ्ट हो गए थे अपने नए कैंपस में, उसके बाद IISc के साथ थोड़ा कनेक्शन मेरा कम रहा था. आज मैं दोबारा यहाँ पे हूँ और उसका पूरा श्रेय सर देयर वी गो, यू चेंज योर सीट्स, प्रोफेसर टंजन को जाता है, जिन्होंने एक ऐसा मुद्दा चुना जो राष्ट्रीय तौर पे महत्वपूर्ण भी है और वैज्ञानिक तौर पे डूएबल भी है। सो सर थैंक यू वेरी मच। और आप जब मैं यहाँ आ रहा था आप लोग तालियां बजा रहे थे, वो थोड़ी कम बजी थी, मैं चाहता हूँ सर के लिए थोड़ी ज़ोर से बजा दी जाए, क्योंकि मेरा मानना है कि विज्ञान हम सब कर रहे हैं, सब यहाँ साइंटिस्ट ही हैं, और बाय हार्ड कोर मैं अपने आप को भी एक ऐसा ही वैज्ञानिक मानता हूँ, और आपको कुछ वही आज बताने का प्रयास करूँगा। अभी शुरू का कुछ हिंदी में, उसके बाद इंग्लिश में शिफ्ट हो जाएंगे। तो आज का जो पूरा प्रयास है, जो मैं आज बताने का ट्राई करूँगा, वो ये कहूँगा कि आयुर्वेदिक जो ड्रग्स हैं, जो हम क्लासिकली सेंचुरियों से यूज़ करते आ रहे हैं, पूरा इंडिया या इंडिया के बियॉन्ड भी, जब हम बात करते हैं हर्बल मेडिसिनल सिस्टम की, तो सिर्फ आयुर्वेदा ही नहीं है। आयुर्वेदा जैसे 63 मेडिसिनल सिस्टम और हैं जो पूरी दुनिया में इस्तेमाल किए जाते हैं। हम आयुर्वेदा के बारे में ज़्यादा जानते हैं क्योंकि हम भारतवासी हैं, हमें वो थोड़ी समझ अच्छी है। पर उसके अलावा चाइना में यूज़ होता है, ताइवान में होता है, अफ्रीका में बहुत ज़्यादा होता है। लगभग दुनिया का 2/3 हिस्सा ट्रेडिशनल मेडिसिन को यूज़ करता है। उसका प्राइमरी कारण कॉस्ट होती है, क्योंकि ये सस्ते होते हैं, सर्व सुलभ होते हैं, और इनकी पहुंच उस जगह तक होती है जहां सिंथेटिक ड्रग्स नहीं पहुंच पाती। तो उसके बारे में थोड़ा मैं आज बताऊंगा कि उन औषधियों का हम वैलिडेशन कैसे कर सकते हैं, कहाँ कैसे गैप हैं। और जब मैं बताऊंगा उससे पहले जहां मैं आजकल काम कर रहा हूं, मैं काम कर रहा हूं पतंजलि रिसर्च इंस्टीट्यूट में, जो हरिद्वार में गंगा के किनारे है। आप लोग जो भी थोड़े से इंटरेस्टेड हों, आप सबको ये आमंत्रण है, आग्रहपूर्ण निवेदन है कि अगर जब आपके पास वक्त हो तो प्लीज़ हमारे पास में ज़रूर आइए। आपको वहाँ देख के बहुत अच्छा लगेगा कि विज्ञान की एक शाखा आयुर्वेद भी है और उसको फॉलो अप किया जा सकता है। तो मैं अपना टॉक शुरू करूं, जिसकी आज टाइटल है आधुनिक रसायन जैव की शोध आधारित नई आयुर्वेदिक औषधियों का विकास, इंग्लिश में ऑफ कोर्स ड्राइविंग आयुर्वेदिक ड्रग डिज़ाइन स्ट्रेटजी फॉर मॉडर्न कीमोबायोलॉजिकल एनालिसिस अप्रोच। उससे पहले मैं एक छोटा सा 4 मिनट का वीडियो आपके सामने प्ले करना पसंद करूंगा, जो उस संस्थान का इंट्रोडक्शन है जहां मैं काम करता हूं, और वो इंग्लिश में है। तो अगर आपकी अनुमति हो सर, कैन आई प्ले द वीडियो?
With the mission to establish the knowledge of Ayurveda and Yoga, Patanjali Research Institute started its journey in 2017 with its formal inauguration by the Honorable Prime Minister of India, Shri Narendra Modi ji. The term Ayurveda is derived from the Sanskrit words Ayur, meaning life, and Ved, meaning science or knowledge. Thus, Ayurveda translates to knowledge of life. Based on the idea that disease is due to an imbalance or stress in a person's consciousness, Ayurveda encourages certain lifestyle interventions and natural therapies to regain a balance between the body, mind, spirit, and the environment. Ayurveda has its origin in Atharvaveda, the fourth of the 4 Vedas. Signs of Ayurveda were documented by the great sage Maharshi Charak in his world-famous treatise Charak Samhita around 5000 BC. Another great sage, Maharshi Shushruta, who is revered as the father of surgery, has authored Shushruta Samhita, the compendium on surgery. Patanjali Research Institute, or PRI, is engaged in the research and development of Ayurvedic drugs. PRI aims to establish India as the launchpad for the growth and development of Ayurveda and become a prototype for the rest of the world, with an aim to become a global research institute on traditional Indian medicinal systems. PRI has 3 divisions. First, Drug Discovery and Development Division. Second, Patanjali Herbal Research Division, or PHRD, and Ayurveda and Sanskrit Division. With the goal of achieving global scientific acceptance for Ayurveda, the Drug Discovery and Development Division, besides engaging in R&D for Ayurvedic drug development, backs up the scientific rationales of Ayurveda with mechanistic studies. Drug Discovery and Development Division is an interdisciplinary research unit with 5 departments: chemistry, in vitro biology, in vivo biology, microbiology, and clinical research. At Patanjali Research Laboratories, the modern scientific rationale behind ancient wisdom of Ayurveda is being deciphered and validated using cutting-edge technologies as per reverse pharmacology concept, with a resolute target to establish Ayurvedic medicines as evidence-based medicines. Patanjali Herbal Research Division, PHRD, is involved in taxonomical identification and documentation of the endemic medicinal flora of India and world. In order to address the growing awareness and acceptance of Ayurveda, Patanjali has taken up the ardent task of offering the World Herbal Encyclopedia. In this compendium, nearly 1 million vernacular names of medicinal plants in more than 1650 languages of the world are being incorporated. The handmade canvas paintings of 35,000 plants are being included as illustrations in the encyclopedia. This work will get compiled into nearly 100,000 pages with approximately 600,000 references and citations. It will provide detailed analysis and description of more than 60,000 medicinal herbs preeminent in more than 2,000 ethnic groups across the world. Patanjali Research Institute has a herbal garden housing more than 1,000 medicinal plants. The institute is working towards amalgamating modern science with traditional knowledge of Ayurveda with the target of Sarve Santu Niramaya, meaning let all be free from illness. Patanjali Research Institute is taking major steps towards developing general and scientific awareness regarding Ayurveda and Yoga in India and across the globe.
Thank you very much everyone for your attention for these 4.5 minute video, and from this I will now switch towards my presentation and try to give you a gist of what we do, how we do, and what is the long-term plans that we have for the institute. Now am I visible there? Of course. So as you already saw in the video that PRI, Patanjali Research Institute, was inaugurated by our Prime Minister Shri Modi ji in 2017. And by looking at that, we are a very, very young institute. It's just more than 5 years here. So we believe that we are still in our infancy. अभी भी हम अपनी शैशव अवस्था में हैं और तरुण अवस्था की तरफ जा रहे हैं. That means we are entering into the age of maturity and would be able to do what we want to do with a bigger goal of the institute. So these laboratories was inaugurated in 2017. These are ultra modern labs which have been set up to bridge the gap between the modern science, modern practice, and our ancient medicinal knowledge, Ayurveda and Yoga. These are probably one of the kind, literally state-of-the-art facility to conduct Yoga and Ayurveda research in the whole world. Top-of-the-line equipments, globally trained workforce, energize the research operation on a daily basis. The mission for our institute. When you talk about any institute, there are always three major components: mission, vision, and strategy. How are we going to do that? So the vision that we have, the mission that we have, that we aim to improve the health and quality of life through research and innovation. हम अनुसंधान और नवाचार के माध्यम से स्वास्थ्य और जीवन की गुणवत्ता को सुधारना चाहते हैं. और हमारा जो विजन है वो ऐसा है कि हम जो एंशियंट क्लासिकल नॉलेज है मेडिसिनल प्लांट की और जो रिच हिस्ट्री है हमारे पास उस सब को एक साथ एक बल्क में लेके साइंटिफिक मेथड्स के फनल के थ्रू बाय फॉलोइंग वेरियस स्टेप्स हम फाइनली एक जगह पहुंचना चाहते हैं जिसको हम साइंटिफिकली वैलिडेटेड एविडेंस बेस्ड मेडिसिनल सिस्टम कह सकें. मतलब कि जो औषधियाँ, जो हर्बल कंपाउंड्स, हर्बल एक्सट्रैक्ट जिस पे हम काम कर रहे हैं उनकी एक्सेप्टेबिलिटी सिर्फ और सिर्फ भारत में ना हो, भारत के बाहर भी हो, उसके जो एविडेंसेस हों वो वर्ल्ड वाइड एक्सेप्टेबल हों. जैसा कि आज हम एलोपैथिक सिस्टम की दवाइयों को मानते हैं क्योंकि उनके केस में हमारे पास एविडेंसेस होते हैं. तो आयुर्वेदा सालों से प्रैक्टिस हो रहा है। समहाउ ये एक जनमानस की भावना की तरह है कि हम ये मानते हैं कि ये दवा काम करेगी, बुखार आएगा गिलोय खाएंगे, अश्वगंधा खाएंगे, ताकत आएगी, बुखार जाएगा। पर वो ऐसा हो कैसे रहा है, उसका एविडेंस क्या है, वो एक बहुत बड़ा गैप है। उस गैप को भरने का काम PRI की टीम करती है और वही हमारे विजन का एक हिस्सा है। और इसको करने की हमारी स्ट्रेटजीज क्या है, हमने इस पूरे एपिसोड को 4 हिस्सों में बांटा। एक इनिशिएशन, ट्रांजिशन, एक्चुअलाइजेशन एंड एक्सलेंस। तो इनिशिएशन लगभग 1995 से 2000 के बीच में शुरू हो चुका था PRI के बारे में सोचना, और पूरा एक प्रोसेस तभी शुरू हुआ था कि दिव्य फार्मेसी को लगाया गया, एंशिएंट विजडम को और विजनरी लीडरशिप के साथ इसमें आगे बढ़े। ट्रांजिट हुए, नए प्रोसेस लगाए, नई दवाइयों को बनाया और वर्ल्ड क्लास इंफ्रास्ट्रक्चर को खड़ा किया, और ये लगभग 2017 के बाद से शुरू हुआ। अब तक लगभग 700 करोड़ रुपए का इन्वेस्टमेंट पतंजलि की तरफ से PRI में है, जो इक्विपमेंट्स, लैंड्स, बिल्डिंग, एम्प्लॉयज़, उन सबको मिला के है। एंड बायोलॉजी एक एक्सपेंसिव साइंस है, उसके कंज्यूमेबल भी काफ़ी महंगे आते हैं, एनिमल ट्रायल्स की कॉस्ट काफ़ी सारी होती हैं। उन सबको मिला के एक जो लीडरशिप का सपोर्ट है वो रियली वर्ल्ड क्लास है, और उस लेवल का काम शायद और कोई अभी इंडिया में नहीं कर पा रहा है जिस लेवल पे पतंजलि पहुंच रहे हैं। उसका लार्जली थॉट प्रोसेस है कि किस तरीके से चीज़ों को हम आगे बढ़ा पाएँ, और ऐसे बहुत सारे काम बगैर फाइनेंशियल सपोर्ट के नहीं हो पाते हैं। उस सिलसिले में पतंजलि की जो लीडरशिप है उनका सपोर्ट वर्ल्ड क्लास है। लगभग अब तक इतना सारा इन्वेस्टमेंट है और इसको आगे लेके पूरी टीम चल रही है। अभी हम जहाँ पे हैं इस पूरे स्टेज में, एक्चुअलाइजेशन वाले स्टेज पे हैं, जहाँ हमारे पास इनर्जाइज वर्कफोर्स हैं, इग्नाइटेड थॉट्स हैं और क्वालिटी डिपेंडेंट डिलीवरी है। और जो हमारा फाइनल गेम है, जो हमारा ध्येय है, वो है एक्सीलेंस, जहाँ पे आयुर्वेदा का यूनिवर्सल एक्सेप्टेंस हो। तो इस तरफ हम आगे बढ़ रहे हैं, और अब उस स्टेज पे पहुंच चुके हैं कि जो विज्ञान है आयुर्वेद का, जो विज्ञान है हर्बल मेडिसिन का, उसकी एक्सेप्टेबिलिटी इंडिया में और इंडिया के बाहर भी आनी थोड़ी थोड़ी शुरू हुई है। पर जब हम बात करते हैं रिसर्च की, तो एक बेसिक एक्रेडिशंस हैं, एक स्ट्रेंथ हैं जो एक रिसर्च इंस्टीट्यूट के पास होने चाहिए। और ये एक नया इंस्टीट्यूट है, 5 साल पुराना इंस्टीट्यूट है। तो जो भी रिक्वायरमेंट्स हैं, जो भी लीगल रिक्वायरमेंट्स हैं, जो भी रिसर्च रिक्वायरमेंट्स हैं, उस सब पे भी PRI ने काम किया है। और आप इस स्लाइड पर देख पा रहे हैं कि जो हमारी लैब्स हैं उनमें 3 लैब्स NABL से द्वारा एक्रेडिटेड हैं, जहाँ डेडिकेटेड क्वालिटी कंप्लायंस है। हमारी लेबोरेटरीज़ का DSR एक्रेडिशंस हैं। 300 से ज़्यादा वैज्ञानिक यहाँ काम करते हैं। टोटल वर्क स्ट्रेंथ PRI की 300 की है, जिनमें आई थिंक 200 के लगभग PhD हैं, 50-60 पोस्ट डॉक्स हैं जो ग्लोबल एक्सपीरियंस के साथ हैं, जो बाहर से पोस्ट डॉक करके वापस आए हैं और आजकल PRI में काम कर रहे हैं। एक फुल फ्लेज्ड रजिस्टर्ड एनिमल हाउस है, जो आयुर्वेद इंडस्ट्री के लिए, आयुर्वेद डिपेंडेंट रिसर्च इंस्टीट्यूट के लिए एक बड़ी बात है, क्योंकि रिसर्च इंस्टीट्यूट जैसे IISc है या NCBS है, ऐसा वहाँ हो पाना बहुत कॉमन है। पर आयुर्वेद डिपेंडेंट सेंटर्स में एनिमल एक्सपेरिमेंट के लिए एक डेडिकेटेड एनिमल हाउस का होना भी एक बड़ी चीज़ है, जो PRI में है, जो एनिमल एथिक्स के द्वारा ड्रिवन है। एक डेडिकेटेड हर्बेरियम है जो न्यूयॉर्क बोटेनिकल गार्डन से एक्रेडिटेड है। रिसर्च लैब्स हमारी बायोसेफ्टी लेवल 2 के तहत गवर्नड हैं, जिनको इंस्टीट्यूशनल बायोसेफ्टी कमेटी रन करती है। डेडिकेटेड कमेटीज़ हैं इंस्टीट्यूशनल एथिक्स कमेटी की, जो ह्यूमन क्लिनिकल ट्रायल को रन करते हैं। एक अच्छी बात यह है कि पतंजलि हॉस्पिटल नेक्स्ट डोर है, जो PRI से ही जुड़ा हुआ है और 350 बेड का हॉस्पिटल है। और अप्रूव्ड आयुर्वेदिक मेडिकल कॉलेज है, जिसमें PG 8 सब्जेक्ट में हैं, जिनके MD स्कॉलर्स भी इसी रिसर्च में कंट्रीब्यूट करते हैं। और लगभग 2500 doctors across India are on the roster of Patanjali, and the daily footfall of the patients are 15,000 in a day, and that keep on compiling in a daily basis. And we have something called Patanjali Electronic Medical Record, PEMR. The total patient population at PEMR right now is close to 70 lakh patients, because that have been happening for last 10 to 15 years, and different diseases, different treatment methods have been given to those patients, and data have been compiled in the same way. So you already saw in the video that PRI has a major these 5 segments: drug discovery, development, herbal research, painting, herbarium, ancient documentation, and yoga research. Other than these 5, we have 4 more sister institutes: Patanjali Organic Research Institute, Central Lab at Patanjali Ayurveda Limited, New Product Development at Patanjali, and Patanjali Hospital and Medical College. All 9 of these are the ones that actually take the research forward for Patanjali Research Institute. So this is at Patanjali Research Institute, Drug Discovery and Development. And these are just a glimpse of our infrastructure. As I said about the top line equipments and technology, that whatever is needed for the research, we have it in-house at PRI. Be it a LCMS, be it a UHPLC, be it a very fancy IBSc system for keeping animals, or setting up the in vitro biological experiment from confocal microscopy till going up to high content screening. All of that is in-house at PRI for conducting the research in a time-dependent manner. And drug discovery and development has 5 divisions, as we just discussed about it: in vitro biology, in vivo biology, herbal chemistry, microbiology, and clinical research. Under in vitro biology, we have various cell lines of human origin, extensive cell biological support, genomics and microbiology, and recently we have moved into nano Ayurveda, where we start to make the nanoparticles from herbal extracts and try to find whether the efficacy of those particles get better when you make them in a smaller size. In vivo biology, in our animal house, we work with rats, mice, guinea pigs, rabbits, zebrafish, and recently we have also added C. elegans. In herbal chemistry, there are 3 major components: analytical chemistry, which has LCMS, GCMS, UHPLC, FTIR, and SFC. Extraction chemistry has SCFE, where we use liquid carbon dioxide to extract the major compound from the herbs, and a different co-solvent that we use as well. And we also have a pilot plant, where we also make the prototype formulations in form of tablets, syrups, or powders. Microbiology team has actually works on bacteria, fungus, virus, and super infections. And clinical research, of course, take care of clinical trials, clinical surveys, and pharmacovigilance. Now, how we do the work that we do. So there are 2 ways of doing it. When we are working on Ayurvedic medicines, we have close to 1,000 different formulations which are described classically. So one could take one formulation at a time and try to see which disease it might be working better and try to find out its mode of action. That would be called a drug-driven approach. What we do at PRI, we do other way around. We actually take the drugs that we have and try to bin them in different diseases compartment. For example, we bin the drugs that might work better for inflammation. And under inflammation, you have rheumatoid arthritis, IBD, asthma, bronchitis, COPD, or psoriasis. And that way you try to find out what might be the good combination which might give a better clinical outcome. And that way we can bin them in the 5 major sections: inflammation, metabolic disorder, cancer, neuroscience, and infectious disease. And the numbers you see next to it is the number of the formulation that we have worked for in last 5 years in these areas. So these are a few formulations which has been developed from PRI, and I just put this slide just to show that Ayurveda has come out of the concept of chutney and choon. That is not like taking something and making a powder and giving it out. The modern formulation types are now also available in Ayurveda. We have tablets, we have syrup, we have powders, we have sprays, we have ear sprays, we even have band-aid. I mean, there anything that is possible which is there out in as in medical technology, it's now available for Ayurveda as well. So what I'm going to talk today. So this is so much about introduction. The focus that I have been working and my team has been working significantly is the concept of reverse pharmacology. And I call this reverse is the new way forward. So what's the reverse pharmacology? So in a pharmacology terms, I mean, when you're working in a pharma industry, what you do that you find a new compound which gets synthesized by medicinal chemists, make a whole library of that, and do some in vitro experiment, do some in vivo experiment, and go to the clinic. And eventually, if everything is all right, in 10 years' time, when with a close to $1 billion cost, you might have a drug that would hit the market. That's a normal pharmacological process, which is expensive and has a lot of bottlenecks, and compounds may fail at any stage because there are a lot of things which are not very well understood in clinics. On the other way, when you're working with medicines which are derived by tradition, where those medicines are in clinical practice for several centuries. For example, Ayurveda. When we know that those drugs, those plant extracts have been showing a good efficacy for last so many centuries, then we know at least that these drugs are safe and they are also efficacious. So now what is left that you can go back from there? That's the reason I'm calling the word reverse pharmacology. You get reverse from there and you then start going from man to mice to molecule. Earlier you were going molecule, mice to man. So the reverse path is actually faster, it is cheaper, and is more likely to succeed because you already have a clinical proof of concept that these drugs work in a given paradigm. It's just we have to find out how they work. And when we talk about how, our body doesn't know that what kind of medicine we are taking. It may be allopathic, Ayurvedic, homeopathy, Siddha, whatever that might be. When the drug goes inside the body, it's a biochemistry that takes over. The drug is going to engage the specific target, receptors, enzymes, many things possible, and that engagement gives a treatment benefit. And when we talk about treatment benefit, there's one more point that I would like to add. I strongly believe, and Ayurveda also say the same thing, that there is not a single drug that cures a given disease. कोई ऐसी दवा नहीं है, कोई ऐसी औषधि नहीं है जो बीमारी ठीक करती है. औषधि और औषधियाँ सिर्फ हमारे शरीर को ताकत देती हैं बीमारी से लड़ने के लिए. मतलब हम खुद ही अपनी बीमारियों से ठीक होते हैं, औषधियाँ उसमें हेल्प करती हैं. इसीलिए एक एक औषधि का अलग अलग लोगों पे अलग टाइप का इफेक्ट आता है क्योंकि हम अलग टाइप से बने हुए हैं. तो सेम थिंग वर्क्स इन आयुर्वेदा टू, कि जब हमारे पास एक प्रूफ ऑफ कॉन्सेप्ट है कि ये दवा इस तरीके से इतने सालों से काम कर रही है, मैं खाली ये पता करना है कि वो दवा किस टारगेट को किस तरीके से एंगेज करेगी और किस तरीके से उस दवा से लड़ने की शक्ति हमारे शरीर को मिलेगी. एंड दैट्स अ होल कॉन्सेप्ट PRI हैज़ बीन वर्किंग ऑन. So this is the balance that we talk about. So we have modern science, what we call technological advances. That means getting new type of microscope, getting a very fancy DNA sequencer, getting a very good quality scanners, which can detect things faster, better, and cheaper. And then we have modern science, where we talk about new drugs which are being tested in clinic. So you have a clinical practice which is very well evolved. Unfortunately for Ayurveda, that evolution has been rather slow. For last 200 years, we have not got a single new drug from Ayurveda. We have been just talking about what is mentioned in our classical books. We know this formulation with this kind of extract will do this kind of thing, which is mentioned in 10,000 year old book, 2,000 year old book, and that's all it has been. Ayurveda has not grown with the same speed that modern science and modern medicine has grown up. And that's a challenge that we want to take it on. We want to balance these three acts. We want to take Ayurveda, modern science, and modern medicine together and set up this golden triangle of drug discovery approach. In Hindi, संतुलन में ही समाधान है. That means we balance in all three together and move forward and try to find out how things would be showing an effect as they should be doing that. So all the work that we do, we get them published, and in last 20 odd months, there are more than 75 papers in peer-reviewed high impact factor journal. The total impact factor is close to 370 with average impact at 4.5 and so. It has been rather difficult to publish a lot from Ayurveda, largely because the acceptability with the peer reviewers are low. But we tried a lot. We have been pushing. Things start to move slightly better now. So we work on many themes. There's a lot of work the whole team does. They're like, as I said, 300 scientists in 5 different segments. So they do a lot of things. But the main themes that we have generated a lot of good data. I just trying to show some of them. The first one is talking about insights into classical Ayurvedic medicine using modern techniques. This is for mucormycosis. This happened just after COVID, where what we call black fungus disease. It happens because the patient who took a high amount of steroid, they become susceptible for this fungal infection. So in Ayurveda, there is a nasal drop called Anutela, which is described 2005 years before by Rishi Charaka. And he said that this oil would work very well for nasal infections. It's mentioned in his book. And we then try to find out, can we see if that formulation would have any effect on mucormycosis. And I'm very happy to report that we found a very significant change. We could see that the fungus growth, which is coming, which causes mucormycosis, can be actually decreased in a dose-dependent manner by Anutela. And we have published recently 2 papers on the same theme. Second, we work on development of a new formulation which is derived from the ancient text through the technology that we have it now. So mixing it ancient with modern and try to find what we can get it. And there are a lot of medicines that we have worked upon. And again, that comes about re-establishing the science of nanoparticles. And that's where we talk about 2 components. One is Bhasma. Bhasma, I'm sure some of you would be knowing it. Those are very fine particles either from metal or from a mineral, and the size of those particles are lesser than 200 nanometer. So technically they are all nanoparticles which have been synthesized several hundred years before. And then we try to find out how those particles would have a therapeutic advantage. Second process, we work with Dhupan. So Dhupan is again a sort of air sterilization technique which is there in classical text, which has come from yagya. So when you put herbal compounds in a flame, they generate some fumes. Those fumes has a very tiny nanoparticles, which indeed has an effect on different kind of microbes, and we have published that as well. So we also work on those to find out what are the science behind those ancient practices. And third part, or the fourth part, which is rather close to me, because I come from a background of drug discovery. I worked for pharma industry for 11 odd years and found out that, every time you do something new, you find something good. In Ayurveda, that has not happened much because, as I said, we were working on the old techniques, old medicines which have been described in books. So what we do new? Can we find some new medicines from some new plants? Or can we find a medicinal use of a plant part which has not been described in past? For example, if there is a plant where the roots are described to have a medicinal value, can we look for the leaves, seeds, or the whole plant for the same? That way we would be able to find out some new drugs from the same source. And if that is the case, how do we do that? So my team works that on very, very high details, and I will talk about some of it today as well. And the last one, learning from the other system of medicine for Ayurveda. That means the system which we talk about here is allopathic system, where there are a good deal of advancement have been made for different kind of formulations. For example, we have entry-coated tablets. That means the tablets which will not be dissolved in the stomach, they would be dissolved in the larger intestine, which helps to deliver the drug right at the site of action. So we learn from that, and I think the first medicine from Ayurveda, which is entry-coated, has come from PRI as well, where we have got a formulation which have been coated with the help of those compounds, which will make it stable in the stomach, but it will get dissolved very well in the larger intestine. So we learn from that too. So today, what I'll talk today in terms of science would be ashwagandha compounds, ashwagandha oils, and if the time permits, a little bit about something about the pain. So this is the first part, Vidania somnifera, ashwagandha. I'm sure all of you would have heard about it. Can I have a raise of hand who knows ashwagandha? Well, everyone, that's good. So ashwagandha, Vidania somnifera, is a popular herbal ingredient of natural medicines, and we have been working on it for quite a while. And what you see on this screen on the right hand side under the red circle are the roots of ashwagandha, which are the part which have been recommended in our classical Ayurvedic test as a medicine. That these are the one that works. So when you talk about ashwagandha, if you have used it ever, you have used either the powder of the ashwagandha root or the tablet coming out from the root of ashwagandha. That means root are the medicinal component of the whole plant. The question comes that why not other part of it? So the whole plant has, this plant is ashwagandha plant over here. They are the flowers, they are the fruits, and they are the seeds. When you have compounds present in roots, similar kind of compounds are also present in other part of the plant as well. So why not look into it? And roots are expensive, they take time to grow. You can't keep on harvesting because once you take the root, obviously the plant is gone. So how about we look for the aerial part of it? And that's where PRI start to look into it. First thing, we looked at the seeds, and we tried to find out what seed might contain and how we can do something like new drug discovery from the old medicinal plant. So if you see the raw material, as I mentioned here, the seeds, we did something called supercritical fluid extraction. So those of you who may not know that, supercritical fluid extraction means that you take carbon dioxide, which is gas, and you compress it at a very high pressure. At a pressure of close to 350 bar, it converts into a liquid. And then that liquid we use as an extractive vehicle. That means you take a powder of the seed, put it in that high pressure liquid carbon dioxide, and whatever the compound present which can be solubilized in that liquid would get solubilized there. You filter it out, take the filtrate and release the pressure. So what will happen? Carbon dioxide will evaporate back in the air, and you are then left with the most purest form of extract possible, which is totally green because CO2 won't be left there, and you would get a good quantity of the item as well. And since this is very polar in nature, you get mostly oils. So if you take the oil from, for that matter, coconut seeds, or from sesame seeds, or from sarson seeds, you will get good quality oil from the same method too, but it's too expensive to use. But for medicinal use, it can be done. So we do have this plant at PRI, and we did this and try to find out whether is there any oil from ashwagandha seeds. And what we found, it indeed had 16% of oil. 16% oil is a very high oil yield, and if we took those oil sample and do a GCMS and GCFID measurement, which is to look for the what are the chemical composition, we found it has good quality fatty acids. Some of the fatty acids which are known to have an effect on skin, and some fatty acids have been shown, the synthetic fatty acid of the same type have shown to have a good effect on psoriasis. So that we thought, okay, if that is the case, can we then test these oil which is coming from ashwagandha seeds for anti-psoriatic activity? And that we actually did. And then my team from microbiology start to look the effect on antimicrobial and antifungal activity as well. Second thing we did with the whole plant extract, where we did a hydromethanolic extraction, and we generated an enriched extract which contains a single compound called vidinone. And now when we talk about single compound, that comes to the same argument that I have been trying to explain to many people, that single compounds are good. They do show some very good activity. But at the same time, you might want to have many compounds which are naturally occurring there too. But times when you have to show a very intricate biology, it's sometimes good to show the biology coming from a single compound too. So we did that with the whole plant, and I'll try to show you some data. So this is the machine for supercritical fluid extraction, where you put this and get this out. Quickly, I'll show you some data for anti-psoriatic effect. Now, there are going to be some data heavy slides. So if there's something which I'm going too fast, and if I miss some of you, please raise hand, I'll repeat it. I also stammer in between, so I might be missing some point too, so pardon me for the same. But after the meeting, I'll be happy to take the query if anyone has it. So this is the GCFID graph that shows the different kind of fatty acids present in this oil. So you can see palmitic oil, stearic oil, stearic acid, oleic acid, linoleic acid, and nervonic acid. Out of this, linoleic acid seems to have a very good effect on skin. So we got very excited by that, and we then started to set up the model for the same. So this is the data directly from animal experiment. I had to compress the slide, so I'm just showing the data that came from the animal, where we generated psoriasis in the ears of the mice, and by inducing with a compound called TPA, which induces psoriatic-like inflammation at the ear. And in those ears, this oil was applied, and animals were also fed with the oil. So it was given orally and topically. And later those ears were harvested, and histopathology was done. What you see on the top left is a normal ear, and what you see at panel C. Okay, let me get some, just one moment. Laser would be good here. Here. So these are the diseased animal, and you can see the thickness of the ear has gone up. And also the hallmark of psoriasis called hypertrophic keratinosis, that also happened. But when WSSO, which is Vidania somnifera seed oil, was applied topically and fed orally in a dose-dependent manner, it actually decreased it, which we later published in Biomolecules and showed that it did indeed does what we actually thought about it, and it also did it in a very dose-dependent manner, and it also goes through the inflammatory pathway. These oils also decrease the TNF-alpha and IL-6 level, which are critical for maintaining the steady state of the disease. Now, antimicrobial and antibiofilm effect against Salmonella enterica. So this is done by the team microbiology led by Dr. Swati Haldar. There she did that, the growth of this bacteria was inhibited by this oil in a dose-dependent manner with the IC50 close to 5.78 milligram per ml. And it also reduced the mortality of the bacteria by the same oil in a dose-dependent manner. It controlled the swimming and swarming of the bacteria, as you see over here, untreated versus treated, untreated versus treated. And it actually disrupted the cell wall of the bacteria, because you could then measure the total efflux coming from either from potassium or nucleotide efflux, and in a dose-dependent manner, WSSO actually have this one modified. In addition, it also affects the biofilm which is formed by the same bacteria. It affected the initiation of biofilm, it affected the maturation of the biofilm, and it's also affected the matured biofilm, which got completely eradicated. And you can see those photographs here too, that each red dot is a single bacteria. As the dose of this oil was increased, you start to see the biofilm was going away. This we again later published in Phytomedicine Plus. And the third component was looking for Candida. So Candida becomes a slightly bigger challenge for us. Candida aureus is actually in pathogen that is there in developing world in startup base. In 2004, if you see, that's how the world looked like. Only these two locations, there was more amount of this infection. 2014, it started to increase. It was there in India a lot. 2017, and now 2018. It's there all over the world at this moment. So then how this is increasing and how we can take care of that. So then the thought comes, how about we try this oil as well, since it affects the membrane integrity of the fungus too. So we got a drug resistant Candida auris and try to find that does this oil has an effect. So this is the blank, this is your untreated fungus growing very strongly. This is the known antifungal that doesn't work very well against this fungus. Amphotericin B, which is known to affect it, it's shown to have a good effect too. And here is the oil, which shows a comparable profile as amphotericin B against the same fungus. And then, of course, you can calculate different kind of curves and could show that this drug doesn't work. Amphotericin B works very well too, but at the same time, WSSO in a dose-dependent manner, it affects the life of this fungus. Then we went ahead and procured different clinical isolates of Candida from different countries, from different parts of Asia, Europe, North America, South Pacific, and Asia, and try to find out that, what really happens if we come with this oil. In all of these strains, we were very happy to report that it indeed kill most of the fungus that was growing it up. So that shows that yes, indeed, this oil would have an effect on this fungus, and we can take it forward from there. But how that happened? The how becomes a very important part, which is a main component of the PRA function. We see a biology, but we have to find out how that is happening. And this is generally happening by controlling the membrane permeability by this oil. And as you can see, as the concentration of the oil is going up, the membranes are actually getting compromised. So we then look for the dye that actually gets in. And when you quantify for that intensity, you see in a dose-dependent manner, this oil controls the membrane integrity of the fungus. Please, ma'am. Sure, ma'am. I will remember. It works on the biofilm as well in a dose-dependent manner. And we did some SEM, scanning electron microscopy, and you can see over here, these are the normal matured biofilms, and with the other compound that doesn't have an activity. As you go ahead with this, you start to see the biofilms are being actually broken. And this is what my team called seeing is believing, because they could really see it by scanning electron microscopy that indeed this oil has a good effect against this fungus. And this is just, okay, we have one nice designer in our team, and he happened to keep on drawing fancy stuff. This is from him, that how the Staph aureus, how the Candida aureus get modified by WSO treatment, and it actually affects the ergosterol sequestration and protoblast formation. And later when you put the oil, all the component of the cell wall actually get diluted out, and that becomes very disrupted porous cell membrane of the fungus. So this is so far for the oil I talked about. And if there is a question, I can take it now. 10 minutes. Okay. All right. I'll take a seat there. So here we talked about now whole plant extract of ashwagandha, and we try to find, does it have a compound that we are looking for. The name of the compound that we worked is Vidinone, and we got an enriched extract of ashwagandha, and we call them wine vice, which you see right here. And it did many good things. This was done during the COVID time, and we did a lot of experiment with this extract, and this enriched extract showed a very good phenotype. So first I talked about Vidinone, and this was at the time of COVID. So in 2020 January we start working on it, and we start to do a first molecular docking, as everyone was doing, and we found that this compound Vidinone was sitting very well at the interaction site of ACE2, which is the human host protein, and RVD, which is a part of the SARS-CoV-2 virus, and it does pretty well, and we did some simulation to find it is stable there too. Then later we went ahead to do a biochemistry. We got the both protein procured and did ELISA, where we could see that both of these interaction can be inhibited by this compound in a dose-dependent manner with the IC50 of 0.33 nanogram per microliter. From there, then we moved ahead and to find out that what we can do more on it. This is just to show the chemistry that we could purify the compound so well that this whole extract, this is the extract peak, and this is the one which is a purified compound. So this is really nice and very stable as well. Now, how do you test the zebra? How do you test the COVID experiment at the time when there was lockdown? Because our labs were open, but we could not do many experiments. Largely, that COVID virus doesn't affect rat and mice, because God has not given them this ACE2, which is specific to human. The other thing that we could do is to develop some transplanted animals, xenografted animals. So we chose to work with zebrafish, and we generated some sort of humanized zebrafish. So we took zebrafish. Fish has something called swim bladder, which is like a bag, which is an air sac, and which is sort of orthologous to our lungs, but those area doesn't have some amount of good cells. So we took human lung cells, A549 cells, and we injected them in the swim bladder, and we hoped that these cells would home in at that area. And what you see here, these are the cells which actually got stuck to the swim bladder of the fish and started to remain there for quite a while. And then we realized, okay, this might be a good model to move ahead with it. In these animals, then we injected the SARS-CoV-2 spike protein. And that spike protein induces the changes in these animals as you would see in the clinical conditions. We started to see fever, we started to see inflammation, we started to see kidney damage, and also the way the fish started to behave. The behavior was also compromised. So this whole model was set up to measure the effect of ashwagandha compound Vidinone in this model, because we didn't have anything else at that moment of time. So this is for measuring fever. Now some of you might also think that fish is a cold-blooded animal. their body temperature keep on changing as the water they are swimming in, and we are warm-blooded. Our temperature is always constant. So how would you measure it? So for that, a new device were actually formed, where the fish was kept in 3 different chambers at 3 different temperature, and these chambers were interconnected, like here, 23, 29, 37, and then you introduce the fish right in the center. So the temperature of the fish would be driving the point where this fish would like to spend the maximum amount of time. And on the top we put a camera and did a video recording for 3 minutes and try to find the number of seconds fish spend time in each of these chambers. And this time becomes a parameter to measure how much was the fever. So when we had a disease conditions where SARS-CoV-2 spike protein was given in those humanized fish, most of the fish was spending time over here at 37 degrees Celsius. And when they were treated with ashwagandha extract, they actually shifted back over to the normal temperature zone. You can look at this data here in this data as well, where in a dose-dependent manner, these are 3 doses we have taken, the amount of time fish spent after 3 days of treatment or 6 days of treatment in a time-dependent and a dose-dependent manner, we could find this compound indeed works very well, which we later published in Drug Design, Development and Therapy. Then we moved towards looking for another fungal disease and try to see does it have an effect there too, and we were very happy to see the similar kind of profile what we see for this fungus, and could see indeed ergosterol content in the fungus was actually controlled by ashwagandha whole plant extract. That we published in PLOS, Neglected and Topical Diseases last year. So that comes right into the whole story of ashwagandha, and I ran through the data just to save time. Sir, I have 5 more minutes. Now I'll come to the second part, and I'll promise to be a bit more faster. This is about Ayurvedic intervention for pain. So in pain, there are a lot of different kind of formulation that we get from allopathic system. Ayurveda has some of it, but still nothing has been actually come in form of the concrete formulation. So at PRI, we generated some combination and we try to find out in which pain area they might work. So pain, probably all of you know, come from either like joint pain come from osteoarthritis or rheumatoid arthritis or ankylosing spondylitis. And these are the formulations that we're talking about. So we took 5 different formulations mixed together. These are classically described in these books, and we have mentioned here too. These all 5 of them together come in form of a single formulation. This formulation now we have coded for PNG, and that formulation then start to show a good effect too. Likewise, we do chemistry first, and this is the compositional analysis of PNG on HPLC to find out what it contains, and we make sure that every batch of this formulation contained at least these component and bit to a fixed limit too. And this is just to do some cell biological experiment to find that this formulation is safe to the cells, and it also control different cytokines which has a role to play in inflammation and also in pain. IL-6 right over here, in a dose dependent manner it decreases. IL-1 beta that also decrease in a dose dependent manner. And then we set up 2 animal models. First is the osteoarthritis model of rat, where we induce something called monosodium iodoacetate at the joint space of the animal, which causes osteoarthritis. This is a detailed plan that we can talk about too, where 3 different doses of the same formulation was given in a dose dependent manner. One parallel group was kept for indomethacin, which is an allopathic drug for the same treatment too. And the second model was set up for surgery-induced chronic constriction injury model called CCI, where one nerve in the animal was exposed and it was sutured for some time to cause the injury of that nerve. Again, the same formulation was used, but in parallel, gabapentin, which is an allopathic drug for pain, was tested in parallel too. And here comes the data. So in osteoarthritic model, a PNG has a very strong analgesic property. So you measure pain by 2 different forms. One is called hyperalgesia. Hyperalgesia is the way when you start to feel pain for something that normally causes pain. And then you do something called allodynia, where you start to feel the pain for something that doesn't cause pain normally. For example, a little bit of cold or heat, that's normally not causing pain, but the diseased animal start to respond to it faster. So that we took it on, and we measured that in both of these models, PNG in a dose-dependent manner has a very nice recovery of the pain sensation in those animals. In CCI model, we did cold allodyna, thermal hyperalgesia, tactile allodyna, and taking it all together, it shows that PNG indeed in a dose-dependent manner has a very strong analgesic response. We didn't stop there. We actually then went ahead, since we're talking about OA, we went for radiography. So we took the X-ray of the joint of the animals and scored them for something called a KNL score, which is a clinical scoring for looking for the pain and the joint problem in adults, human. And the same thing was done, and as you can see, PNG in a dose-dependent manner also decreased that scoring. That means the structural change that was happening at the joint were also getting better with the treatment of this formulation. And then our histopathology team jumped in. They took the animals and then they started making different slices of bones and the cartilages. Taken together, that you can see the inflammation score decreased significantly. The scoring for osteoarthritis also went down. And in parallel, we also look for an specific cartilage oligomeric matrix protein, COMP, which is a known marker for the pain in osteoarthritis. We measured that in the serum of the animals, and we could find that COMP level, when we measured by ELISA, actually decreased very well, and it was as good or as bad as indomethacin in the same model too. Now, I have done my PhD in ion channels with Professor Matthew, and when I talked about pains, the first thing that triggers is the channels. So there are different kind of channels which are there in our nervous tissue, and they are generally TRP channels, transient receptor potential channels, and there are TRP B1, A1, A1, B4, B1, B4, M8, and A1, and they are responsible for different kind of pain which is causing either from pH, from heat, cold, or mechanical problem. All of them also play a significant role. So in our animal experiment, we actually went ahead to look for the expression level of these systems. And what we found, the gene expression for these ion channels were also controlled by PNG, and again in a dose-dependent manner. This is for TRPV1, TRPV4, TRPA1, and TRPA8. All four of them, which are the significant one, also decreased by PNG. In addition, the RNA expression for P38 MEP kinase and IL-6R, which is the receptor for IL-6, also decrease in a dose-dependent manner. So taken together, we try to find out the effect of this formulation. Now we are knowing how that is getting modulated, and that how has been the key for us, and we have been trying to get this on. Later, our clinical research team jumped into it, and since we have close to 3,000 patients there at Patanjali taking treatment all the time, so there a little clinical study was done. To them, we gave this PNG formulation and also asked them to do the yoga as have been advised to them for taking care of their pain. And this is just a quick data from that study where they did PNG and yoga. And we did a different kind of questionnaire to see their pain rating, their quality of life, and how the osteoarthritis is getting modified in their own feeling. So this was a single-arm cohort study, and these are the markers, just diagnosed. I'll quickly go through this. Total 370 patients were taken for the same study. 306 was the one which were enrolled, and total we got a data for 276 patients. And this effect, the total pain scale, the pain went down in all three different diseases. The quality of life went up in the two different scales. That means the patients were feeling lesser pain and their quality of life was improved by the treatment of PNG and also with the yoga. And at the same time, when we are looking for the osteoarthritic pain, that also decreased significantly by the treatment either by yoga and along with the PNG, and the stiffness that we talk about, that also decreased. So pain went down and stiffness was also low in the patients who were taking yoga and PNG together. And the difficulty in walking also becomes low for all these three conditions. that comes to the last part of me, that way forward. So what we want to do it forward, and the evidence I try to show some part of it. We have been working very significantly on the same, that the key consideration for developing scientific evidence-based Ayurvedic medicine, the biggest challenge that we find is to get a right amount of starting material. That the plants that is growing in Bangalore versus the one in Haridwar or in MP or in Andaman, same plant would have a different biological response. To find it out that what is the really correct one and it is the right plant at the first place, one need to set up something in process. And that's the one challenge we are feeling at PRI. We might need to do some sort of DNA barcoding or try to find some plant markers so that we will get a correct raw material or starting material to show some activity. And the second thing that we talk about, importance of scientific evidence-based claim substantiation for Ayurvedic medicines. That's what we have been working on, and I try to show you some of this data, and there's a lot more in the public domain. You can go to PRI website and find it out there too. But the idea there is to have a enhanced global acceptability, align Ayurvedic rationale with the theories of modern science with modern scientific techniques, and demonstrate the conceptual robustness of the ancient science that we have been practicing. After all, seeing is believing. And the mission that I talked about, we come back to it, that we really feel about Sarve Bhavantu Sukhina, Sarve Santu Niramaya. You have been a fantastic audience. Thank you very much indeed. This is my acknowledgement to the team that have supported. Swamiji and Acharji have been the guiding light and has been there for all the funds that we talk about it. Their thought process have been really remarkable. Team PRI, Team Patanjali Ayurved, Divya Pharmacy, and Patanjali Yoga Peeth. So thank you very much. If there are any questions, I'll be happy to take it. Anutel.
So there, the drug efficacy starts at milligram level as compared to amphotericin, which is in micrograms, right? Is that a problem when the concentration is so high to give it to people at the right concentration?
Clinically, it's not a big problem because Anutela classically have been described to give 4 drops in each nose.
Anutela, the
Ashwagandha oil, huh?
Where you had this active compound, right? With
Right.
anosome. That also worked in milligrams, like 50 mg per ml.
Right, that's correct.
Whereas
too.
amphotericin works in micrograms.
Microgram.
Right?
But again, the doses for that would be higher as well. And that's one of the area Ayurveda need to really work upon too. The amount of drug given from Ayurveda is actually high. For example, a normal painkiller you give like maybe 100 mg to 200 mg. Ayurveda one goes to like 2 tablets thrice a day.
No, but when you say Ayurvedic tablet, that's the whole mixture of different things. But in case of your Vidanosome, it's actually a specific compound, right? If I understood
No.
it correctly.
There it's an oil that's coming from.
It's an oil.
It's an oil that's coming from the seed. So that oil can be kept in a gel cap, and the gel cap can be given as a medicine. So that is the next part that eventually we will get it going.
Yeah, and then the last one. So PNG. Again, it was a mixture of compounds, and
Right.
you showed that there is
5 of them.
Yeah, so ellagic acid is actually something which blocks RNase L, which is important for inflammation. So I was wondering if other compounds have also been specifically tested for particular inflammation markers, right? And
Right.
yeah.
That has been done, and I think I'll share the paper with you, which have been already published. But thank you.
Yeah, the last slide spoke about one of your goals being to demonstrate the conceptual robustness of ancient science. Now, what exactly are the concepts that you are referring to here? And if it includes, say, the theory of doshas, what is the basis to first suppose that it is a robust concept?
Okay, so I'm not talking about doshas per se. What I'm talking about, the robust concept is that you have very well described herbal components as drugs, as medicines, which are robust, which have been practiced for several centuries, right? It's just a matter of validating by giving scientific evidences, and that's the aim that we're trying to go for. Doshas are again very phenotypic in nature. There is a different team that deals with it. But robustness, here what I mean, just about showing that, for example, ashwagandha, all of you know, going to do this. But how we have to find and convince others outside India too, that's the reason it works. Did I answer the query, sir? Thank you.
Just 1 minute. I'll just come to you. I'm here.
Okay, of course.
Now, because he has brought a very important thing here. So, Anurag, what I gather from your talk is that, you are looking like a reductionist approach, the Western way. And you are trying to claim it as that you are giving a scientific evidence. But the whole Ayurveda is not about getting into the compounds, right? It is a holistic approach. For example, if you take water, if you break it down to oxygen and hydrogen, and you put hydrogen and oxygen, it's not going to give you how the water works in the body. And so I think we are missing a point here is that, we are supposed to do that how Ayurveda formulation, whatever people have taught, they work, but not getting into nitty-gritty of the compounds and things, right? Because ultimately, I would say that whatever you have talked about is that Ayurveda inspire drug discovery, not Ayurveda per se.
Chairman sir, with all due permission, what I would like to say, this is about Ayurveda formulation only. The PNG that I talked to you, this is just the formulation which are described in Ayurveda, and we are showing how they are working. We are not talking about a single compound. The single compound just been used as a marker to identify the right amount of formulation. For example, if you get a tablet of crocin, you need to know it has paracetamol and you want to know how much it is. But here, when we're talking about this formulation, what you see here, this is just a chemical signature of that formulation. So that when we get a new batch of the same formulation, we can compare. But we are not isolating single compound.
I'm not doubting it. This is what you have extracted using some way.
No, it's not extracted. These are, sorry to interrupt you here, sir. These are the classical formulation. For example, if you see Mahabhat, Vidwanshan Ras.
You will get only these 5 compounds?
This is the 5 compound, not 5 compound, 5 formulations which are described in classical thing. For example, Ras Tarangini is the book that is from 12th century, if I'm not wrong, where you have Mukta Shukti Bhasma, which is actually a calcium powder coming from the shell of coral or I think the seashell. So this is described there. We are just mixing them together to show that this combination has a good efficacy, and here is the preclinical evidence and here is the clinical evidence. This part what you are thinking, this is not to purify any single compound. This to just show signature that this exists in this form, in this formulation.
I agree totally. What I'm trying to say is that the sum of this may have you are able to get down to 4, 5, or say 6, or even a single compound, active compound, which may work, and they also may work by inhibiting some, say, receptor or something. But what I'm trying to say is that that may work. And today we know that if you look at these deworming or things, whatever people have described in the Ayurveda, they work by taking out single active compounds. The Brahmi, if you take the Brahmi compound, it works. But ultimately, when the single compound works, the body actually develops some kind of different resistance and things. But when you take the formulation, it works differently.
I agree.
That is where he was talking about the dosas, dosas and things. So the different individuals behave differently, how they work it. I think that is where the Ayurveda, the way things are looking, is different from the. What I'm trying to say is that whatever you have to looks like more of like Ayurveda inspired drug discovery than actually the Ayurveda person.
I beg to differ you, sir. I would want to say the same thing again and again.
I agree. Yeah. Definitely. But what I'm trying to say is that as a person who believe in Ayurveda, in local health things, this is, I also work on Ayurveda from last 15 years. We just identify a compound which can cure epilepsy, the single compound from pomegranate was given to Ayurveda for the neurological. We identified the urolithin works by inhibiting VDEC1 now, which is in the patent. But what I'm trying to say is that, but if you take the pomegranate per se, it is completely different ball game than the, you take a urolithin 1, the compound which we just identified. Is a completely, when it comes to the individual level, at the individual level, how you behave taking pomegranate is different from the individual taking a single compound like urolithin 8. So that's what I'm trying to say.
Sir, you are saying it right, and you are saying exactly what I want to say too. So thank you for echoing my thoughts. But this is exactly what we're doing. We're trying to show that. All right.
Thank you for a very interesting talk. I am so glad that very sincere attempts are being made.
Thank you, sir.
To ensure that there is some hardcore evidence in the traditional practices that we are making.
Right, sir.
My quick point is, are you following this up with classical level 1, level 2 or clinical trials where a large number of humans are involved?
Right, sir.
Blindfold. Maybe it may not be a good idea, but I would use that maybe the established traditional allopathic Western system of clinical trials. Has it been done?
Absolutely, sir. We are doing it right now. There is a clinical research team which has now 50 people. Half of them are MD doctors, and their job is to actually conduct those trials. In next 18 months or so, you would be seeing more data coming out. Those are randomized controlled, placebo-controlled trials which are happening. In Ayurveda, we now made placebo tablets too, which look and feel like the normal herbal tablet, but they don't have that compound inside it.
But then
so we will be comparing it head to head.
Then there is a slight dichotomy there, because if you are going to look at only formulations and then do this, then how are we able to track it to the original molecules? For example, in classical
That's what sir is
allopathy,
saying.
that's the
Not
point
to,
that
so
Upendra
he's saying not to go to the molecule. Let's go towards the formulation. And in clinical practice, if the formulation shows an efficacy, that's fantastic.
But is it repeatable, because the plant from different regions will have different
Right. So
signatures?
that's the reason these chemical testings are must, because then you can define what are the phytocompound present and how much amount. And that's a route what Indian government now allow herbal medicine to take as a phytopharmaceuticals, where you define that each herb would have 3 markers in this much concentration. And Patanjali is now working into those lines where we will get the drug license from allopathic system for herbal drugs. In US, it's called botanicals. The same rule that you show at least these compounds are present in a single herbal extract. And again, sir, it's a herbal extract, not a purified compound.
Thank you, sir. I think integrative medicine is now a reality, so it's a good idea to mix East and the West.
Of
Thank
course,
you.
sir.
Thank you
very
for a
much,
very
sir.
good talk.
Thank you, sir.
I would like to just add, this is for this audience only. So one of the most used Ayurveda product is Chyawanprash. So if you look at the classical text, it's approximate. You take this palmful of, say, amla, then you take it with the ghee and honey, and this is how really medium thing is taken. But however, if you look at it, depending on the thing, there are more than 2,500 variety of amla in India itself. And so which one, where you are taking, how much you have to take, there is a huge variety. There are around 150 formulations in the market at the moment for Chyawanprash alone, with the addition of the tea. Now, which one you will take it, Dabur or something?
Patanjali.
The Dabur, when we have taken it, believe me, Dabur also, when we have tested, it is very easy to test with the drosophila itself. Right. Actually, Chyawanprash increases the, what we call as the rasayana, improves everything. But in the market, we have tested it. Only 4 Chyawanprash varieties could sustain the viability of drosophila. All other drosophila die. So I'm just trying to say is that there is lots of potential, but I think it's a huge.
In the interest of time, I think we should stop.
Sir, I'd like to add one thing. Sir, for just for one comment. Chairman, sir, we did some experiment with Chyawanprash as well, where we did on zebra fish and could see the inflammatory state of the fish get modified when they are fed with Chyawanprash. And those fish were alive when they were given Chyawanprash. Of course. Thank you.
I'm an Ayurvedic patient associated with some of the molecular biology research also. The thing is, exactly the methodology what we are approaching, the either reverse pharmacology or in the drug discovery, structure targets, is it really a right model for validation of Ayurveda or for the publication? Because since 30 years I'm working in the field, almost 89, when we go for the publication as an Ayurvedic physician, when we want to adopt the principles of Ayurveda as a whole, like what sir was saying, dosha dhatu mala theory, that if you go for the ethical committee in front of the nationalized institutes or with collaboration, corporate things are different research. When we go for the real research, it may be very, very challenging for us as a researcher to go, either clinical research or basic research also, either clinical scientist or basic scientist also. How we might have overcome, because this really we should appreciate. We have to go for more publication. And also, where is the principles of adopting? It is not that egg or hen, but we have to come out with the, if you go, even Sean May and Zakir Sanaza, we were talking, but they want data. Until they allow us to give accumulate the data, they won't, we can't publish. So what is the, another thing is, the last question is, when we are selecting that extraction methods, why you want to specifically select one specific extraction only, rather than, so many methods are there for the extraction. When we talk of that oil, why you are, how we can assess that extraction
For extraction, the best way forward is the one which is green in nature. Green means that there is no carryover of a solvent. So if you use methanol or ethanol, they might carry over along with the extract too. So the best way forward is water or liquid CO2. But not all the compounds will commit out in those methods. So you might need to mix a little bit of methanol to get the maximum amount of recovery. And that's where the chemistry comes in the picture. So we take in a plant material and do a multiple extraction in different solvents and do an HPLC to find out where we get the max and where we get the stably max. That means every time you do it, you get the same amount of it. And then later we evaporate and try to remove all the solvents possible. So you don't predict, you do the experiment.
We'll go for the tea. We'll discuss there. Just only 2 minutes. We would like to give a gift to Anurag for saying wonderful talk. And thanks to Rajiv also. No, it's fine. Please, Anurag.
Thank you. I'll come this side too. Thank you very much. Very nice. Thank you. It was very nice.
And one for the chairman.
One for the chair, of course.
Thanks to you.
Thank you, sir. Pleasure.
So we have tea
We
and
have
coffee
tea, please
here,
come
please.
over there.
Anurag, I need to go. I need to attend a conference
Okay.
tomorrow.